In the heart of Jerusalem, researchers are hunting for the answers to aging and brain decay.
Alzheimer's is a neurodegenerative disease, meaning that there are nerve cells, neurons in our brain that are the key functional unit of the brain that are dying. When you look at the individual, you have memory loss and cognitive impairment and mood disorders that are slowly kind of taking away the essence of the person in front of you. It's really devastating to see.
And an aging population means there are far more cases.
There are estimates saying that 1 out of 3 elderlies in the United States are diagnosed with Alzheimer's at some stage. You look at a textbook on Alzheimer's, it seems like a lot of the mechanism is already figured out. But when you try and translate that to the clinic, it seems like something is missing.
But at the Edmund and Lily Safra Center for Brain Sciences, Professor Naomi Habib is finding out exactly what is missing.
Our goal is to understand mechanisms of aging-related pathologies, and specifically Alzheimer's disease. We're trying to understand how does, on the most basic cellular molecular level, how does the disease progress from a healthy brain to advanced Alzheimer's disease. And our goal is to find these early triggers of the disease so we can find effective therapeutics against them.
We're very interested in looking at cells that are actually aging. We get donations from live brain tissue from surgeries, and we can grow this brain tissue of a human in our incubators as well. So we have the chance not only to look at cells, but to look at actual human cells at different ages from different patients. And then we have the same advantage here, we can manipulate those cells.
And what makes this lab unique is the ability to do those experiments at the smallest scale.
Now we can actually look at individual cells. And then later in the computational analysis, we can figure out who these cells are and build maps of the whole brain. So how is this done? The key point is our ability to actually isolate different cells, capture them, and tag the molecules of each of these cells individually.
The cells that we see in this map, they come from hundreds of different individuals. Using this diversity, we can now try and put all of these donors along some disease timeline, or to try and learn some disease timeline. And we are finding that we have different of these trajectories leading to different disease outcomes.
And that is uncovering the answers to many long-standing questions.
Some people were claiming that Alzheimer's is an accelerated form of aging, meaning that if we all continue to age until the age 150, we will all get Alzheimer's. But in fact, what our maps show is that there are different paths the cells in the brain can take. One of these paths is where all the cells together are changing their functions, leads to Alzheimer's. And the other path leads to the slower decline that we associate with more healthy form of aging.
And from the answers in Habib's lab, a path to a cure.
There are people that have pathologies in their brain, but they don't have the clinical symptoms. So these type of resilient individuals, we believe, are really the key to find a cure. So we're measuring the brains of specifically these individuals, comparing them to people who have Alzheimer's, and trying to find what are the natural mechanisms of resilience.
And from there, a possible cure for millions.